Today we are going to talk with respect to micropipette. These are micropipettes. Micropipettes are basically a laboratory instrument which have been used to pipette out and withdraw a liquid sample, okay, from one container towards another container. There are three different kinds of a micropipette which have been placed over here. With respect to each one is differing with respect to the volume, how much volume they can take in the liquid and how much volume they can withdraw in the particular liquid. So over here you can see different micropipette which have been placed. Along with that, there are different micro tips which are available with respect to a particular volume which have been selected over here. So we are going to study about how to hold the micropipette. Before moving ahead, how to hold the micropipette, we are going to understand what exactly the micropipettes are, what's the role of each of the micro parts are there, and we are going to see how exactly we are going to work it. So this is how exactly micropipettes are. Okay, so micropipettes are the laboratory instrument which is going to take a selective volume, okay, and is going to withdraw from one container, transfer that particular volume from one container towards another container. With respect to this particular micropipette, as you can see over here, the range, how much volume it can take in over here is from 0.5 to 10. So beyond 10 microliter, it cannot take up the volume, or below 0.5 micrometer, it cannot take up the volume. Okay, so you can take or you can selectively take up the volume within 0.5 to 10 microliter only. Same goes with respect to another which is of 10 to 100 microliter, okay, followed by that which is of 100 to 1000 microliter. Okay, so as I've stated, so these three are the micropipette. Apart from it, there are different kind of micropipettes with respect to those particular volume. Okay, now as you can see over here, this all micropipettes, let's select this particular micropipette. Okay, this micropipette has already been set or has been kept at the higher volume which is mentioned with respect to this particular micropipette, that is 100 to 1000. So it is already always been kept at higher volume over here. Okay, if you try to keep it below 1000 microliter, or if it's basically below 100 microliter, it's going to damage the micropipette. So always the micropipette, whenever it's been kept in the resting position, that means whenever it is not been used, then it has always been placed at higher volume over here, so that the pressure is not been applied onto the spring which is held over here, and which might cause a damage to this micropipette and create an error, okay, while you selectively take out a particular volume over here, as the damage has been created with respect to micropipette. So always remember, whenever you are done with the work, keep the micropipette at the higher volume with respect to that particular micropipette. Okay, so we'll see with respect to micropipette how the working of the micropipette should be carried out. Now with respect to this higher 100 to 1000 microliter, we are going to use this micro tips. So according to differing microliter volume, different kinds of micro tips are available. So with respect to this higher volume, we are going to select this micro tips. Okay, before going ahead, we'll understand how a specific kind of a volume has been selected. Like for example, if you want to withdraw or 500 microliter or 800 microliter, so you have to specifically set the micropipette at 800 microliter, whichever desired volume or how much volume you want to take it out. So if you want to set, if I want to withdraw 800 microliter, so I'm going to set this micropipette at 800 microliter with the help of this knob. Okay, can you see this knob? Okay, you just have to turn this knob. Okay, I'm turning this knob. Now it is set to 800 microliter. So can you see a red mark over here, a red pointer over here, which is pointing, it is 800 microliter. If it is moving, the red mark or red pointer is moving up or bottom of this 800, then it will be something 800 point something over here. Okay, so make a point, this pointer, red mark pointer, is always with respect to the particular marking of a precise volume. Okay, so now the micropipette has been set at 800 microliter. So I could withdraw exactly 800 microliter from the micropipette. Okay, now how exactly withdrawing will be carried? Okay, so for this particular thing, I will firstly I will place the micro tip onto the micropipette. Okay, this is holder where micro tips are in place. So I am going to place micro tips onto this micropipette. Okay, now as I have set it at 800 microliter, so it is going to take 800 microliter, is going to pick up the volume that is 800 microliter only, not above to it, not below to it. Okay, for aspirating the volume or a fluid from the given sample, okay, you have to place the micropipette upright at 90 degree in position. Okay, followed by that, now there are two presses. One, this is single press, okay, and followed by there is one more press, that is second, this is double press. So always apply to aspirate a volume a single press. Okay, if you aspirate or if you applied a double press, it is going to pick up the volume, or is going to take up the volume which is higher than to your required one. Okay, so always apply a single press. We are going to hold the pipette in this particular way. Your 4 fingers should be lying over here. Okay, while your index finger is lying close towards this particular arm of the micropipette. To press this button, you are going to use your thumb. Okay, by applying pressure, you are going to press this particular button. Okay, as you apply a pressure and press this particular button, okay, followed by that you are going to dip your micro tip, okay, something around 2 to 3 depth from the surface of the liquid. Okay, and then you are going to take up the solution. Now to take the solution, you need to release the pressure, okay, which you have applied at the top end, okay, over here. So slowly release the pressure. Okay, as you are done slowly releasing the pressure, okay, now I'm going to place it, I'm going to withdraw this particular volume into another container. Again over here I need to apply a pressure to withdraw the fluid from the micro tips of the micropipette to another container. Single press, no double press, something like that. Okay, this is how exactly the working or handling of the micropipettes are being done over here. As you are done with it, you need to discard the pipette. Now discarding of the pipette, you need to do it, okay, you don't have to hold it over here to remove the micro tips. Okay, you can discard the micro tips, okay, by just pressing this particular knob which is present over here. Okay, as it has been pressed, can you see the one end of the one end, okay, is of the micropipette is moving ahead, touching the micro tips, and is giving pressure to the micro tips to move it out from the micropipette. So you have to press it over here and you have to remove the micro tip from the micropipette. So this is how exactly the working of the micropipette has been done and how it has been handled over here. As you are done working with your micropipette, you need to release the pressure which you have or to given to set the particular volume by again moving to 100 volume with respect to that particular micropipette, that is 1000. Okay, done with it. Now you can taste it. Thank you.